Thank you so much, Ludwig, and thank you to the organizers for inviting me here. It's been a real pleasure to be here. Great conference and always interesting to visit Jerusalem. So I'm going to start with an apology that is probably going to be one of the more technical talks. So if you were in the Schlafstunde before, you will sink deeper into it right now, I think, unfortunately. But I will start by so I'll talk about two methods. One on testing ancestry for direct ancestry. I'll explain in a second what I mean by that. And then a second method that should be number two, not number one repeated, which is using the length of mixture tracks to date admixture events. So the first method, the one for testing for direct ancestry, was motivated, the development of that method, by a research project looking at a sample from the Clovis culture. And just to remind you of what the Clovis culture was, it was the first unique culture that you had in North America, appearing about 13,300, 400 years ago or something like that. You find the first artifact. It's characterized by the stone tools that I'm not an expert on this, but I'm told that it's characterized by a certain way of chipping first from one end and then from the other side, and then from one side then the other side, to make these stone tools. So Clovis culture suddenly appears, you find it all over North America, and then it disappears again. So people have for a long time been discussing the central questions of where did the Clovis people come from, and where did they go? Are modern Native Americans descendant of the Clovis culture, or are they not? And so my collaborator, that many of you will know, Eske Willerslev, managed to get hold of a sample. So Eske was supposed to be here, but he's too busy doing this kind of stuff, so he couldn't come after all here. But I'll talk about a little bit about this anyway. So the sample was from this Antic boy, a sample from that was found in a mountain in Montana, and was deposited about 12,500 years ago. And it was supposed to be clearly identifiably associated with Clovis culture artifacts. And the question then was of course this Clovis individual, the Antic boy that was presumably from the Clovis culture, what modern people was this individual related to? And then we can use the usual type of things that we're doing today, using, for example, a free statistic, outgroup statistic here, so we can make a map of who is the individual related to. And the more red you are, the more closely related you are to those individuals. So we could see that people that live in Central America and South America today are most closely related to the Antic boy. And you have seen mixed trees before in this conference. Here's another one of them. You have a group here with the Carajana and Mayans. These are representative of indigenous people from Central America and South America, and then you have the Antic sample sitting right there. So it's compatible, it's clearly closely related to certain modern Native Americans, and it might be compatible with actually being an ancestor of modern Native Americans. But how do you determine that question? So the question we want to address was this: can we best describe the relationship between reference individuals that we're looking at, so these Native American individuals, and the Clovis individual in a tree like this, where there's some unique divergence leading to the Clovis individual? Or is it more as something like this, where the Clovis individual that we have the sample is compatible with being part of a population that's directly ancestral to the population to which the reference individual belongs? So how can we address that question? So usually in population genetics, when we try to address a question like that, we have highly parametric models where we're trying to describe how genetic drifts and other factors they work along a lineage in a tree. So we might start to do various simulations where we model the changes in population sizes along this lineage here to try to address that question. But it turns out we can in fact address it without making really any population genetic assumptions at all. And the way we do that is that we make a model that has many parameters in it that are not really population genetic parameters, but just simply describes features of the data. So we'll make a model that has a number of different parameters. It has the coalescence probability in this lineage, so the chance that two gene copies that coalesce in this lineage. The coalescence probability in this lineage, the chance that two gene copies coalesce in this lineage. And then we'll ask what's the probability of the configuration that we see here in these two individuals, allelic configuration, given these coalescence probabilities, and then given an estimated ancestral configuration. And the whole key here is that if you put in parameters for this entire ancestral configuration, you don't actually have to assume anything about population size or anything else, because it's all captured by this. So the null hypothesis we want to test is that the coalescence probability here is 0. That means that this branch length is 0. There was no coalescence at all in that lineage, so that the closest individual actually falls down there. So not to put you too much to sleep, I think maybe I'm not going to go through some of it, but I actually put these up to show that it's quite simple, that you can write out some very simple equations for these here, and you're laughing, it's actually true compared to anything else we're doing. This is fairly simple. You can write down some simple equations that show what is the distribution of allelic frequencies in terms of some very simple parameters. And then you can do maximum likelihood, you can estimate all these parameters jointly. And then we can get a likelihood ratio test to test if this parameter is equal to 0, meaning that the branch length is 0. And the advantage of this method is that there are no assumptions regarding population structure or population sizes. We don't have to make assumptions like that. And also, for the statistically interested, it's a full likelihood method which incorporates all information in the data. So there's no loss of information. And this busy slide here shows results for the ANSIC board. And the important thing to look at, I hope everybody can see it also in the back, is over here, where we have these parameters that have to do with how much drift was there in the ANSIC lineage and how much was there in the population compared to. And if we get the estimates of how much drift there was in the ANSIC lineage compared to these ones, Mayan and Karatiana, some of the reference population, the estimate is 0. We get dead on that there was 0 drift on that lineage, meaning that it's compatible with actually the sample, the ANSIC boy, being directly ancestral, falling directly on the lineage that's ancestral to these modern South Americans. So using that method, we could show that, in fact, it's likely that the Clovis culture, it didn't die out, but in fact, many, many modern Native Americans, they're direct descendants of people from the Clovis culture. So I'm going to show you a slightly different example now, and I can't really talk about what this sample is because it's not published yet, and I couldn't talk about it. So this is an anonymous sample. Then I'm going to compare to some reference individuals I'm also not allowed to talk about. But it's just to show another example here. Also Native American, that's 8,500 years old. And we do the same test here. And here we get some estimates of the amount of coalescence probability in the closest population. So if we take, for example, the Karatiana, we get much higher coalescence probabilities. We get a likelihood ratio of 4.23. Clearly not directly ancestral of the Karatiana. But we get some much smaller values for these reference individuals. It's almost compatible with being directly ancestral of these ones, but not quite. We can, for example, look at likelihood surface here. We can look at this coalescence probability here. This is a profile likelihood surface, and we get a maximum likelihood estimate here. But it's not much different from 0. So it's almost compatible with being ancestral. So hopefully next time I'll be talking about this anywhere and actually tell you what the sample is. This time I couldn't. All right, so that's the issue about detecting direct ancestry and a robust method for doing that. The rest of the talk, most of the talk what I'll be talking about is how to date admixture time. So it's a problem that occurs all the time. When did, you have a population, it's mixed between several other populations. When did that admixture happen? We have it with the Neanderthals, Denisovans, but we have it with many, many other populations as well. And so my group has devoted quite a lot of efforts into developing theory that will allow us really to get at that question well. So it's motivated, again, by results like this. It's a structure plot or admixture plot. You've seen many of these in this conference. So these are different components. These are different individuals, and some Europeans here, some Native Americans, and some Polynesians. Almost all individuals are clearly, easily categorized as being either European, being Native American, or being Polynesian, except there's some individuals here that cannot. They are admixed. They somehow contain genetic components that can be traced back both to Polynesia, to Native Americans, and to Europeans. And we want to look at individuals like that to say something about when did that mixture between these groups, when did that happen? Okay, so the approach we use for that is to look at what's called admixture fragments. And let me, just to explain to you what that is, just consider this slide first. So imagine that you have a first generation admixed individual. So for example, this could be a human chromosome and a Neanderthal chromosome if it was a human Neanderthal hybrid. So in the first generation hybrid between humans and Neanderthals. Then in the next generation, recombination will break up this segment here, this chromosome. So you get a shorter segment here. Okay, if you imagine that the individual breeds now with some other humans, retains some of the Neanderthal DNA, but it gets broken out by recombination. next 5 generations later, there's been more recombination. So these fragments have become smaller here. And if you go to 100 generations, they become even smaller yet. So the point here is that how long time has passed since admixture, you can read that off the length of these fragments, right? If it's long time ago, these fragments are very short. If it's very recently, these fragments are very long. But what is actually the distribution of these fragments? So we have done quite a lot of work on trying to figure that out. It turns out it's much more complicated than people previously thought. But to a close approximation, the admixture track length distribution is exponentially distributed with this parameter here. So it depends on the admixture proportion, the effective population size of the population, and the time since admixture. An important thing here is that previously people have kind of ignored the fact that genetic drift also contributes to that. That has to do with the way you back recombine 2 fragments to make a new fragment. But that turns out, especially as the mixture time is long, to become very, very important. We can also derive other things. For example, we can derive what's the variance in the admixture proportion. So in the plot I showed you before, there's some individuals that were part Polynesian, for example, but they varied in how much Polynesian they had in them. And we can use that variance among the individuals also to estimate that mixture time. And here we have an expression for what's the variance among individuals in the population in the mixture proportion, which depend on the sample size, admixture proportion, effective population size, length of genome in recombination units, and also the time since admixture. And because it depends on the time since admixture, we can estimate the time since admixture from this by simply finding the right variance. All right, so let me show you some examples where we worked on this. One example here is the Kostenki sample that previously was mentioned. It's an individual that's at least to a certain degree European, could be described as European, but 36,000 years old. And if you look at the admixture tracks there, you'll get a distribution like this, K14, that's Kostenki. So this is a track length, different samples, French, and so on. Oh, I should say of Neanderthal DNA. Important little point I forgot. So the proportion of Neanderthal DNA, the admixture track lengths. And the Kostenki sample here, you can see in average, you see if you look at the little boxes here, it's considerably longer than it is in the other individuals. That's what you expect. It's an old sample, right? So the time since admixture to Neanderthals is much shorter for the Kostenki sample than it is for the other ones. And we can use that to get an estimate of the Neanderthal divergence time, which we got here to be about 54,000 years. Of course, it has large variance around it because there's only a limited number of mixture tracks in an individual, and that creates some variance. Okay, so that was the Kostenki sample. Here's another example, the Rapa Nui. So those are the indigenous people of the Easter Island, where again in a project where Eske Willerslev was one of the people leading the project, we were asking the question, how old what is that mixture? And in particular, is that mixture that we see of Native American DNA in the Rapa Nui, does that predate the first European contact? All right, so did people from the Americas before, in pre-Columbian time, did they make contact to the Eastern Island? The data we had was this data I showed this slide before. These are individuals from the Easter Island. You can see they have some European in them, some of them have a lot of European, and then all of them have some Native American. And from this you should already see that there's a hint in this, because I talked about the variance before. You could see the variance is very high for the European admixture. Some have a lot of European, some have very little European, but the variance is not very high for the Native American admixture. That's compatible with the Native American admixture being much older than the European admixture. Okay, so that's just the variance. But what do the track lengths say? Okay, so we fitted several models, and I should say that when I say we, it's mostly work by Victor Moreno Maya and Ana Malaspinas who are doing this work. So they fitted 2 different models. One in which you did not allow the Native American admixture to be older than the European. That's this one, M2. And one in which you allowed it to be older. And the estimates you would get of how old the admixture were, if you allowed it to be older, would be that the Native American admixture is 21 generations, and the European admixture is 4 generations. If you forced it to be that the Native American could not be old, they both get estimated at 8 generations. But the likelihood that you get by fitting these track lengths, by fitting a model to the distribution of these track lengths, is much, much higher in the model that allowed the Native American admixture to be older. Strongly statistically significant, distribution of track lengths is simply much shorter for the Native American DNA. And this is another way of, or this somewhat busy figure illustrate that. If you look to the left, you might be able to see that the red ones here, the dots are the real data. The purple, the shading and the lines has to do with the fitting of the models. But the dots that sit in here, those are the real data. You might be able to see that the red ones here, the slope is much faster here. That's the Native American DNA than the blue one here, which is the European DNA. This is the track length, this is a count, and this is the length of the tract. So this means that the European tracts here, the blue ones, are much longer than the Native American ones in average. Again, telling us that, in fact, the Polynesian, the Native American admixture into the Polynesians is much older. So from that we concluded, in fact, and there's been some other evidence from that previously, that there have been contact between Native Americans and people of the Easter Island before Europeans. All right. So the last example I will talk about is EPAS-1, which has to do with altitude adaptation in Tibet. So that's something we in my group have been working on for many years now, starting a collaboration about 5-6 years ago with BGI, Beijing Genomics Institute, on this. And what we did was, originally back in 2010, was we identified a locus that was responsible for heritable changes in the production of hemoglobin and red blood cell production in Tibetans. So Tibetans have, by anthropologists for a long time, been described as one of the groups where you have clear evidence of adaptation to the local environment. So the environment at the Tibetan plateau is of course highly hypoxic, low in oxygen. The partial pressure of oxygen is about 60% of what it is at sea level. So every time you get a breath of air, you get only 60% as much oxygen as what you do at sea level. The consequences we all know: you'll be out of breath and so on. You can develop mountain sickness. But after a little while, you will tend to acclimatize, and you will do that by producing more red blood cells. So all of us here in this room that are not Tibetans, we'll be doing that, and we'll end up having quite a high concentration of red blood cells in our blood. Now you think maybe that's a good idea, but it's not really, because it changes the viscosity of the blood. The blood becomes too thick, and that predisposes you to stroke and all other cardiovascular diseases. So there are all kinds of problems, for example, for all the Han Chinese individuals that are part of the Chinese government's policy of moving people up in Tibet, you know, that they're living up in Tibet. They have all kinds of physiological problems. For example, it's difficult for many women to carry their pregnancies to term. They suffer from preeclampsia, so type of hypertension during pregnancy and so on. So that's all well known. But the Tibetans are not doing that. They actually have a lower concentration of red blood cells at high altitude than other people. They somehow manages to survive in that environment, in the hypoxic environment, well, without having to produce so many red blood cells, so they don't get all these negative physiological effects. And we did a selection scan to figure out where there's been unique selection in Tibetans in 2000 and back in 2000, and we found one of the two genes that are responsible for this. But the major gene is a gene called EPAS-1 that we could show doing association mapping in fact controls the hemoglobin concentration in the Tibetans. So after that, we started to do a lot of work on trying to model, because we like models and stuff like that. We started to model how does it work when selection is acting on a variant like that in a population like the Tibetans. And we asked various questions. For example, we were trying to answer the question: was this selection from standing variation or from a de novo mutation? So when humans move out in the world and encounter new environments, do they carry the genetic variation with them that will allow them to adapt to these environments, or do they have to wait for a new de novo mutation to happen before they can adapt? That's an old question, and we thought, okay, this is a good case to look at that. And we did a lot of modeling, and much of what we did points to the fact that, in fact, it's likely selection on standing variation. The distribution of haplotypes in the genome fits best with that. But what we also saw as we were doing that was that none of the fancy models that we developed really fit the data very well. We could, in some sense, reject all of the models that we had. And the problem that we saw was that if you take Han Chinese and Tibetans in this region, they're just simply too differentiated from each other. And that's illustrated by this figure here. So here each row, that's an individual, and each column is a particular SNP, a particular variable position in the genome. If you're white, that means you have the ancestral type. If you're black, you have the derived type. And so the pink one here, those are the Tibetans, and the yellow ones are the Han Chinese. And you can see in the Han Chinese, there's one haplotype that's common, and in Tibetans, there's another haplotype that's common. But the problem is, these two haplotypes are too differentiated from each other compared to what you would expect under any model that has genetic drift and demography and natural selection in it. that the natural selection acting on one or even two variants wouldn't create a pattern just like this. And we developed various models where we're trying to, could we simulate on a various model with selection to get so much haplotype differentiation? It's illustrated by this figure here. So this is simulations on a number of different models of the amount of differentiation you have between the two haplotypes. Okay, so that's the distribution of all the simulated data. And in the real data, it always lies way out in the tail, in the red line here. So we couldn't really explain, we couldn't find a model that could explain the amount of haplotype differentiation. So then we asked, okay, maybe it was gene flow from some other population, but this haplotype only exists, the Tibetan haplotype, in Tibet, and then also you'll find it in Nepal and in Mongolia, and other high altitude adapted populations in the same area. But it doesn't exist anywhere outside that area. There's no related haplotype to it any place outside that area. So we thought, it must be Neanderthal, but it's not Neanderthal, in fact. It has no similarity with Neanderthal. Neanderthal is very, very divergent from this. So we kind of gave up. But then Matthias Jacobsen's group published a paper arguing that there was Denisovan DNA that has been introgressed into East Asia. That it's not only in Melanesians, as people previously said, but this enigmatic group of individuals, we only know them from their genome, the Denisovans, somewhat related to Neanderthals, but not Neanderthals, that they in fact had also introgressed a bit into East Asia. So we then compared to that sequence, that's the sequence up in green here, and it had an almost complete match. So it seemed like this was Denisovan introgressed, that the DNA came from Denisovan somehow into the Tibetans or the ancestors of the Tibetans. So now we wanted to show, in fact, quantitatively that that's not true. That's true. One thing is to show similarity, but we also have to answer the question: could that happen randomly? Of course, we share a common ancestor with Denisovans, maybe 500,000, 600,000 years ago. So maybe randomly there could be fragments like this in the genome with this kind of similarity. So we looked at this problem in a couple of different ways. One way to look at it, just to get you warmed up, is to look at a haplotype network. Okay, so this pie chart here represent a particular haplotype, and the coloring represents different populations. You can probably figure out what most of them are. For example, CHB, CHS here, the yellow ones, that's China. And the pink ones here, that's the Tibetan haplotype, the selected haplotype. And the green one here, that's the Denisovan. So one thing you see is that the distance between the Tibetan and the Denisovan haplotype is 9 mutations. But there's 40 mutations to the nearest human haplotype, which is, by the way, not anything that you find in East Asia. It's not an East Asian haplotype. There's 35 differences between Denisovans and other humans. And an important point here is that that the Denisovan is actually closer to modern humans than the Tibetan is. And that's what you would expect, because the Denisovan sequence is maybe 40,000 years old. So the Tibetans have had 40,000 years to accumulate mutations on that haplotype that the Denisovans don't have. And if you do the calculation, that fits pretty well with the difference here in the numbers, that you should expect an average about 5 more mutations in the Denisovan haplotype. So that all fits very well within integration. We did other things as well, things like using Patterson's D. Many of you that do ancient DNA, you will all be familiar with Patterson's D. It's a way to measure if there has been integration, if you compare, basically is how tree-like is your data. And Patterson's D is measuring that, and it goes between 1 and -1, and the value for this Tibetan haplotype, when you compare with Denisovans and as an outgroup, then it's very close to being -1, so the most amount of admixture that you could find. So usually when we use Patterson's D, we have a genome-wide data, and we can use various block bootstrap approaches to assign statistical significance. We can't do that here, so we have to simulate. So again, we simulate it on the various models. And these are distributions of Patterson's D you would expect if there's no integration on a bunch of different models. And the red line is the observed value. So again, we couldn't really explain the value of Patterson's D without integration. But the last argument we used is then the length of the admixture fragment. So we can look at the minimum length the fragment could have conservatively. And then we could use this kind of logic again and look at the length of the fragment, and then calculate that if it had been admixture, the admixture, our null hypothesis is that admixture had to be older than 300,000 years if it was similar to being ancestral lineage sorting. And we can reject that with strong statistical significance. In other words, the tract is so long that there's no chance it could be shared ancestor variation, because given the recombination rate in that region, that tract should have been broken up by recombination long time ago. It shouldn't be that long anymore. Okay, it couldn't, it couldn't have existed for 300,000 years with that length. All right, so I'll end with that there. So that was our sort of final evidence for the fact that this is in fact admixture, and make sure that I acknowledge all the people that have been working on this, particularly all the theory on admixture tract lengths has really been developed by a PhD student of mine, Mason Liang, who sadly now is going out to develop algorithms for breaking the stock exchange instead of doing genetics. So that's a sad waste of his life. All the people doing work on the Clovis project, lots of people, Morten Rasmussen, Pontus Skoglund, Michael DeGiorgio, many other people, the Kostengi, and then Gunnar Orlando, Thorfinn Cornelius, and Martin Sikora. Already mentioned the people on the Rapa Nui, and of course Eske Willerslev being part of all these projects. And also in particular, I'd like to mention Emilia Huerta Sanchez, who was doing leading all the work on integration in Tibet. Thanks.
We have time for 3-4 questions. Yes, please.
Okay, thank you. Yes, it was wonderful. I had 2 questions. Have you done any comparisons with the Peruvians? Because high in the Peruvian mountains also they have, there's some kind of adaptation. And then also the 54,000 years ago for the contact between the Homo sapiens and the Neanderthal, where did that occur for that population?
So let me take the last question first. We'll all like to know that. So there's lots of debate and discussions about that. Yeah. I think the main hypothesis, the Pilo hypothesis, right as Pilo came out of Africa, they admixed in the Middle East somewhere, and then that's why all populations outside Africa has a Neanderthal admixture. Right. But you see difference in the mixture proportion between East Asians and West Eurasians, for example. So there has people have hypothesized, okay, perhaps the explanation is not as similar like that. But I know many groups are working on that. It's a lot of active research on that. And probably other people can talk about that. People have lots of opinions about this. The other question was the Peruvians. Yeah, so that's another thing that lots of people are working on. We're also working on that right now. There have been a number of studies. We know it's not the same. We know there are certain genes that have been under selection in Peruvians, but it's not the same genes. Because there it's not the hypoxic response pathway that so much has changed. And they actually don't also have the same physiological response. They do have an increased production of red blood cells, but they have other phenotypes. For example, they have the barrel chest phenotype. That's been described anthropologically, and we still don't know if it's heritable, actually. There's no studies showing, okay, is this just acclimatization? You grow up in that environment, you tend to get bigger lungs, or is it actually something genetic? We don't know, because anthropologists have had sort of the tradition of just describing what they see, and somehow implicitly they would assume that it's genetic, but we don't really know for sure. Okay.
Yeah, this is just how robust your admixture dating technique is. If it wasn't one boost of admixture, but it was more continuous in time.
Yeah, so we have results for continuous admixture and also for multiple discrete. We can write down equations like this equation here. It'll take a little while, but this one here. We can write down equations for this also with 2 or 3 admixture events, or for continuous admixture. But at some point you lose your power to be able to distinguish. And I think the admixture tract length you lose, except if you have very, very large samples, or it's in exactly the right time frame, you lose a lot of power to distinguish between that. But we can certainly distinguish between 1 and 2. And we can also estimate roughly times of 2 admixture events if we have a reasonable amount of data, at least in simulations.
So what's your estimate when this admixture with the Denisovans has occurred? Because it would somehow argue if it was already the Denisovans present, they were high altitude adapters, but there's not much technology in high elevation, which goes back to the Pleistocene, so it was really cold during that time, I guess. So would it rather, so I guess there's 2 different hypotheses. It happened also 45, 50,000 years ago, whenever Denisovans came here.
Well, so
it was something that was actually a reminiscent population was still around. So have you, for example, then looked at early modern humans if you find it somewhere, or can your track length tell you when it actually occurred? If it maybe occurred much later than
Good question. So we have all data we looked at, and we have never found it anywhere else. So that's to answer that part of the question. On the age just dated from the fragment length there, the confidence intervals we get is between 10 and 60,000 years for the integration side. We get a maximum likelihood estimate of about 25,000 years or something like that, but it goes between 10 and 60,000 years. So I don't think it's terribly informative. And it's just because we have one track, right? So, you know, we can't do too much of that. You get very wide confidence intervals. But we also, when I was lying slightly before when I said you don't see this haplotype anywhere else. You actually, there's a couple of Han Chinese. If you look at large sample, you'll find a low frequency among Han Chinese. It also exists maybe 2-3%. Those Han Chinese don't seem to be otherwise more related to Tibetans. So there's no evidence that they are sort of recent back gene flow from Tibet. Of course, it could be old and we couldn't detect them. But it suggests to us that the haplotype existed for a long time. And then when people went up to the Tibetan plateau, then selection favored it. And there's another reason why that I think that's a reasonable model, is that there actually been experiments at low altitude on expression. And it doesn't, the gene, there's no change in expression at low altitude of the gene, only at high altitude. So presumably, there's no phenotypic effect. There's no cost at having that mutation at low altitude. But it does give you a benefit at high altitude. So that will also be compatible with it having linked around neutrally for a long time, and then when they went out in high altitude, it was selected. So that's what we're speculating. Of course, we don't know.
One last question, one more question.
So, what more can you say about the contact between the Polynesians and the Native Americans? Is there any if there was a chicken genome that was supposed to
There was a chicken genome. There's some empty DNA that also has suggested this previously.
There's a Native American genome that's supposed to be Polynesian.
Ah.
But, I mean, do you see?
Well, I think the other, no, I think this is the only clear case of contact, I would say. There's lots of debate about Polynesian contribution, perhaps even Melanesian, into the Americas. But I think of material, at least that's published right now, I think this is the only clear genomic evidence. Of course, there's some linguistic evidence, and there are also some crops where that suddenly, you know, you find them in the Americas and they shouldn't be in the Americas and so on. So so there's other indirect evidence, but to my knowledge, this is only direct genomic evidence.
All right, thanks again.
Thank you.